So last time we started talking about proteins. Of course, we looked at the amino acids, 20 amino acids with the different R groups. We looked at how they linked up through the peptide bond. And we also saw, or I talked about how those bonds are flexible. Many of those bonds are flexible and they can, you have a long protein, let's say 200, 300 amino acids, they can fold up into, an infinite number of possibilities. But as it turns out, due to the primary sequence, that is the sequence of the amino acid R groups and specific interactions, they fold up spontaneously into the correct shape most of the time. We saw that with the experiment from Christian Amundsen, where he showed that the RNase molecule, when it was denatured and unfolded, would placed in water, just folded back up and it became functional. And that's due to the precise sequence of the amino acids. And then we looked at some of the interactions that take place between the R groups in water, and the R groups with each other. They can make the hydrogen bonding and the ionic and covalent bonds, interactions with the peptide bonds, and so on. So those are the kind of interactions that occur. But what I want to talk about today is basically what kind of structures are formed. Start looking at what do proteins look like, kind of the three-dimensional structure. In a sense, that's part of what's called proteomics, which is a field of genetics and biochemistry where people try and understand proteins better and what they look like and how they function. And the question is, can we do that? Can we predict from the amino acid sequence? Because in principle we should be able to do that. Can we predict from the amino acid sequence, the primary sequence, what the structure would be? Well, it's not simple at all. There's an overwhelming number of potential interactions. All these different R groups can interact in different ways. And so you have to be able to sort through in a way and find the correct folding. What's the pathway and the most thermodynamically stable conformation that would form? So it's a area of research that's very intense, and I will tell you that it's basically impossible to do. This is a picture of the second fastest, or part of the second fastest computer on the planet. The first is called the Summit computer. It's made by IBM. The fastest computer just came out is in Japan, but this is the Summit computer. It is a extremely high-speed computer. There's 200,000 processors, and it's in a building the size of 2 basketball courts. It's enormous. Of course, it takes an enormous amount of energy. The computer is a petaflop computer, 200 petaflop. That's the term that's used for 10 to the 15th, basically 10 to the 15th manipulations per second. So we have gigahertz, which is 10 to the 9th. That's most of the computers we use. 10 to the 9th calculations per second. A terahertz is 10 to the, or sorry, a teraflop is 10 to the 12th, and a petaflop is 10 to the 15th. And this computer here is 200 petaflop computer. That means it can do 200,000 trillion calculations per second. 200,000 trillion. That's 2 to the 17th calculations per second. Now, with 100 amino acids, a protein of 100 amino acids, it's estimated that there are 10 to the 47th possible conformations. Of course, only one of those is correct. So how could we need to figure out the most thermodynamically stable conformation, taking into account all the interactions with all the various amino acids and their positions and so on? Well, with this computer, it can be done, but only on very small proteins, very small peptides. With an 100 amino acid peptide or 100 amino acid protein, it's estimated that it would take over 100 days of working this computer 24/7 in order to come up with the solution. 100 days of computer time for a 100 amino acid protein using the second fastest computer on Earth. So it is a monumental task, and it's really not feasible at this time. So let's look at the next slide. This shows just the figures of the diagrams of two potential proteins. And you can see the complicated structures that these things can make. Very complicated, but these are the correct structures that these particular proteins can make. So you can imagine how, with a large protein, it would be virtually impossible at this point to calculate, even using the fastest supercomputer, to calculate how these things could correctly fold up. But having said that, I want to talk about a particular individual, a scientist named Linus Pauling. Linus Pauling, let me move to the next slide. Linus Pauling was a chemist. He lived between 1901 and 1994. He died at the age of 93. He died in 1994. If you look at the paper that's on the bottom, the Proceedings National Academy of Sciences, it shows when this last paper was published. It was published in 1994. So the year he passed away, he published his last paper. This man was considered a genius, kind of like Einstein of chemistry. He basically understood the bonds. He was somebody who had an insight, a tremendous insight into chemistry. And he won a Nobel Prize in 1954 for his study. Basically, he discovered the bond, chemical bond, covalent and ionic bonds, and he worked out the conditions of those bonds and published very important papers on that. And he won a Nobel Prize in 1954 for his study of the chemical bond. Interestingly, he also won another Nobel Prize. He won a Nobel Peace Prize. It's very unusual for somebody to win two Nobel Prizes, but he won his prize in chemistry in 1954 for chemistry, his work on the bond, chemical bond, and also the peace in 1963. He was one of the movers in the anti-proliferation movement. You can see on the right-hand side, it's him marching, holding a sign. At that time, in 1963, I won't go into this too much, but in 1963, or in the 1960s, I would say, it was the height of the nuclear proliferation between the United States and the Soviet Union. There was basically an arms race. And they were doing nuclear testing, they were doing testing in the atmosphere, they were doing nuclear testing underwater. And there was a fear that there would be some kind of a all-out nuclear war between the United States and Russia, the Soviet Union at that time. And in fact, we came very close to that with the Cuban Missile Crisis when President Kennedy was the president. You can read up on that. I don't want to go into it too much, but the fact of the matter is that we were probably minutes away from launching nuclear missiles to the Soviet Union. And they were minutes away from launching nuclear missiles towards the United States, literally minutes away. And Kennedy and his brother, who was the Attorney General, who was Robert Kennedy, who was also assassinated when he was running for president, but they were able to get in some kind of secret contact with Khrushchev, who was the leader of the Soviet Union, and somehow calm the situation down. But honestly, we were minutes away. It was right there. And that was a huge fear that there would be a nuclear war, and so there was a huge movement to stop nuclear testing, and he was the leader of that. As a result, he had his passport taken away. He was considered a communist agent. He was ostracized. He was not allowed to go to any kind of scientific meetings because they felt he was anti-American. But in any case, he won the Nobel Peace Prize in 1963 for those efforts. So that was Linus Pauling, a real giant in chemistry and also in the peace movement. But Linus Pauling understood the chemical bonds so well, he started studying protein structures, and as I just mentioned in the previous slide, that we need some high-speed supercomputers to even begin to understand what some of these structures might look like. But as it turns out, he was able to figure out two of the most common structures just by using paper and pencil. He understood how these bonds interact. So he came up with what I have written here, the alpha helix, in 1948. The alpha helix is an extremely common structure on proteins, and he determined that not using computers, but using his knowledge of chemistry and his knowledge of bond energies and his knowledge of how these things can rotate, and also interactions between the peptide bonds. So he was the first one to come up with any structures of what a protein might look like. He came up with the alpha helix and also the beta sheet a little bit later. Let's just take a look at the alpha helix. So here's three different ways of looking at the alpha helix. Like I said, it's a very common structure, and this is referred to as a secondary structure. So the primary structure is the amino acid linked together. The secondary structure is considered small localized regions of folding. And the alpha helix is a classic secondary structure, as is the beta sheet, which I'll show you in a second. So the alpha helix is a right-handed helix. It is stabilized between, so if you look at the left-hand side showing the ball and stick model, it's stabilized by hydrogen bonding between the peptide bonds. That's what I showed you or talked about a day ago or 2 days ago. So it's stabilized by the interactions between the peptide bonds. The R groups, I think, have to be a little bit on the smaller side. But there are 3.6 amino acids per turn. And it's, as I said, it's right-handed helix, and usually it's smaller R groups. But again, this is something that is held together by the interactions between the peptide bonds, and Pauling was able to figure that out just by thinking about it. Here's a picture of a protein, structure of a protein, and later we'll talk about how these structures are generated. This is a protein called DNA gyrase, which is involved in DNA replication, but what you can see here are all the alpha helices in this protein. You can see them, they're drawn as helices. So this is a particular protein with lots of alpha helices, which is a very common structure. The other very common structure Linus Pauling also discovered, and that's the beta sheet. And that's what's shown here. Again, I showed you something like this last time was my drawings, but these things are stabilized by the peptide bonds, and you can see that in the drawing on top. And the way we usually draw this is that the different strands of the sheet, which are held together by the interactions between the peptide bonds, are running in alternate directions. So if the arrowhead, let's say, would be the amino terminus, then you see how the amino terminus is on one side, and then in the next run of the sheet is on the other side, and it goes back and forth. That's the only way that these R groups can line up appropriately. And he of course was able to see that. And here's the structure. Also, I have the structure of another protein. Honestly, I don't remember what it is, but it doesn't really matter. But what you can see here are all of the beta sheet structures in this particular protein. And many proteins have combinations of beta sheets and alpha helices. So very common, another very common structure. So the alpha helix, beta sheet, two common secondary structures. And oh, by the way, I just remembered what that protein is. I'm sorry. It's a caspase. It's a protease that's involved in apoptosis, but it doesn't really matter. The point is the beta sheets. Now, I want to go back and talk a little bit more about the alpha helix, because there's a very particular type of alpha helix which is also very important and something that's important to know about. So this is a structure called an amphipathic alpha helix. It's an alpha helix of the protein. You can see the right-handed helix, 3.6 amino acids per turn. But what's unusual about this helix is that it's amphipathic. And if you remember when we talked about the hydrophobic and hydrophilic interactions, I mentioned the term amphipathic when I showed you the fatty acids. And amphipathic refers to having dual properties, both hydrophobic and hydrophilic, in different parts of the molecule. And the amphipathic alpha helix is such that the amino acids are arranged in such a way that as the helix turns, the hydrophobic amino acids are on one side of the helix, and the hydrophilic amino acids are on the other side of the helix. So the helix has two chemically different sides. Polar R groups on one side, nonpolar R groups on another side. And so these things can interact in water in an analogous way that the fatty acids interact with water. And that is, they will position themselves, a few of them will come together, they will position themselves so that the polar amino acids are facing outwards into the solution, and the nonpolar or hydrophobic amino acids are facing inwards, excluding water. And that's what's shown here on the next one. So here again is the amphipathic alpha helix, and what happens is, with this type of arrangement of the amino acids, what it says up on top is you get intertwining alpha helices. They form a coiled coil. Two of these come together, and they form a coil which is shown in the middle, where you have the nonpolar on the interior of the coil and the polar on the exterior of the coil. So this is a way for proteins to come together and hold themselves together. And in fact, there's one very common type of coiled coil, which is referred to as a leucine zipper. Leucine is a nonpolar amino acid, and in many proteins, what you see, many proteins which have these amphipathic alpha helices, the primary nonpolar amino acid is leucine. So you'll see all these runs of leucines that are turned on one side of the helix. So they refer to them as a leucine zipper because it allows them then to come together. And as it turns out, many DNA binding proteins are made up of leucine zippers. The leucine zipper holds these proteins together. And what's shown on the right-hand side is just a simple diagram of a protein which is bound together through this amphipathic alpha helices binding to DNA. So here you have a secondary structure which causes proteins to come together. Now there are other secondary structures, and I'll just mention, I think just one more, or maybe, yeah, I think just one more, just to give you an idea. So this is another structure called a zinc finger. So you have leucine zipper, you have zinc finger. It's another structure, and again, it's due to specific spacing of amino acids. And so what's shown at the top in green is you have two cysteines together. It says it should only be 3 amino acids apart. It says 7, but that's a mistake. I'll correct that next time. So you have a C at position 3, another cysteine at position 6. Then you have a space of 13. Then you have histidine and another space of 4, and you get another histidine. So you have cysteine, cysteine, histidine, histidine. And what happens is when these are spaced in that arrangement with precisely that distance between them, they fold up in the presence of zinc. And zinc is held in some kind of a coordinated fashion between the cysteines and the histidine. Zinc in this case is a cofactor. So I mentioned cofactors last time as another contributor to three-dimensional structure, and so what cofactors are non-protein elements that contribute to protein structure. So zinc is a non-protein element, a non-amino acid element, but is required to be integrated into that in order to make this zinc finger. And the zinc finger kind of makes a loop coming off of the protein, and you can see that at the bottom. So you have the zinc coordinated, and then you have a little bit of beta sheet structure, and you have some alpha helix structure, and it forms some kind of a loop, little loop coming off of the protein. And these are often involved in nucleic acid binding, either RNA or DNA. So the zinc finger is often very common in DNA binding proteins. So multiple levels of protein structure, and we're kind of moving up little by little. So you have the primary structure, of course, is the linear sequence of amino acids. You have the secondary structure, which we just talked about, small regions of three-dimensional folding. And the classic is the alpha helix, beta sheet, zinc finger is another one, and there's others, but these are the most common, particularly the alpha helix and the beta sheet. And then they form up, these things fold up into larger complexes, which are then referred to as the tertiary complex, which is a larger three-dimensional region of folding. It's more complex. It involves multiple secondary structures. And then, as I have written in green, tertiary structures often refer to protein domains, which we'll talk about not in this PowerPoint presentation, but in the next one coming up. And then there's quaternary structure, which we'll talk about a little bit later. Now, all these kinds of structures are represented by what are called ribbon diagrams. Now, we've already seen these, but I haven't mentioned the word before, ribbon diagram. So these are classic ribbon diagrams. Ribbon diagram showing the helix, ribbon diagram showing the beta sheet. These are secondary structures, and then we have more of a tertiary structure, which is a single protein domain, where they're folding up in a more complex fashion, and then you get an entire protein made of two different three-dimensional structures in a sense. So you have the one on the bottom in yellow, and then the one in the blue on top. And these are ribbon diagrams. Let me just show you some others. These are ribbon diagrams of 3 different proteins. Cytochrome B, which is involved in electron transport. a region of lactate dehydrogenase, which shows a lot of beta sheet structure and also alpha helix structure, and then part of an antibody molecule. This is a part that interacts with some substance that's infecting the body. It could be a virus or it could be bacteria, some other foreign agent, but what's shown here is a lot of secondary structures. Now, what we'll talk about next in the next PowerPoint presentation, which I'm going to show you, is, well, the question is this what proteins look like? The answer is yes and no. Proteins don't look like this. They don't look like cartoons. They're made of molecules. There's lots of energy around them. There's lots of movement. But these ribbon diagrams are a way for us to represent what they look like. Now, they are not just, the information that generates these ribbon diagrams does not just come from, oh, we think these have an alpha helix, we think these have beta sheets. They actually come from experimental analyses of protein structure, and then we represent it like this. They do have the alpha helix and the beta sheet and all these different secondary structures. And so overall, the proteins kind of look like that. Of course, they're not red, they're not green, they're not yellow, but that's kind of the overall structure. Of course, it's not showing the bonding and the interactions and the energy, but it kind of just looks like that. The thing that I want to kind of point out is that proteins are real substances with real shapes, and they're not amorphous blobs. They're very, very specific shapes, and of course each shape has a different function. Okay, so we're going to stop here, but the next one we are going to look at how some of this is done. In other words, how some of these structures are generated.